Hello everyone, welcome back to my channel. I am Dr. Deepika Malik and today we'll be starting with a new topic that is immunoassays. An immunoassay is a biochemical test that measures the presence or concentration of a macromolecule or a small molecule in a solution, typically biological substances such as proteins, peptides, hormones, drugs, and even certain pathogens, through the use of an antibody usually or an antigen sometimes. This method relies on the highly specific interactions between the antigens, substances that induce an immune response, and antibodies, which are proteins produced by the immune system in response to antigens. The molecule detected by the immunoassay is often referred to as analyte. Analytes in biological liquids such as serum or urine are frequently measured using immunoassays for medical and research purposes. Immunoassays rely on the ability of an antibody to recognize and bind a specific macromolecule known as analyte. In immunology, the particular macromolecule bound by an antibody is referred to as an antigen, and the area on an antigen to which the antibody region, known as paratope, as you can see in the diagram, binds is called an epitope. In some cases, an immunoassay may use an antigen to detect for the presence of antibodies which recognize that antigen in a solution. In other words, in some immunoassays, the analyte may be an antibody rather than an antigen. In addition to the binding of an antibody to its antigen, the other key feature of all immunoassays is the means to produce a measurable signal in response to the binding. They can emit radiation. The technique for this is known as radioimmunoassay, or produce a color change in a solution, technique known as ELISA, or fluoresce under light, technique known as immunofluorescent assay. Determination of antigen concentration in a sample requires production of a standard curve using antigens of a known concentration. The concentration of antigen in a sample can then be calculated using the optical density, as you can see a graph placed towards the right side. Labels. Immunoassays employ a variety of different labels to allow for detection of antibodies and antigens. Labels are typically chemically linked or conjugated to the desired antibody or antigen. These labels generate a measurable signal that indicates the presence or quantity of the target molecule in the sample. The type of the signal depends on the specific immunoassay method. Following is a table where on the left side you can see the immunotechnique and then the type of label that is present in the technique and the type of signal that is generated in that technique. First one is radioimmunoassay. The type of label used for this technique are radioactive isotopes, and these isotopes emit radiations that can be detected by a radiation counter. Immunofluorescent assays use fluorescent dyes, and these dyes emit light of a specific wavelength when excited by a corresponding wavelength of light. ELISA, that is enzyme-linked immunosorbent assay, uses enzymes as labels, and these enzymes catalyzes a reaction that produces a detectable color change. Now let us discuss these techniques one by one in detail. Radioimmunoassay. It is one of the sensitive immunoassay techniques which helps in measuring the concentration of specific antigens or antibodies in a sample with the use of radioactive isotopes or radioisotopes as labels. It is an in vitro type of antigen-antibody interaction, which means experiments or processes that are conducted outside of a living organism that measures the presence of analyte with very high sensitivity. The classical radioimmunoassay methods are based on the principle of competitive binding. Radioactivity emitted by bound antibody-antigen complexes can be easily detected using conventional methods. Requirements: radio-labeled antigens or antibody. A known quantity of the antigen of interest is labeled with a radioactive isotope. Commonly used radioisotopes in radioimmunoassay includes iodine-125 and tritium. The labeled antigen is referred to as tracer or hot antigens here. Second is specific antibodies. They are required in smaller amounts than antigens. Third requirement are the unlabeled antigens, also known as sample antigens. They are also called cold antigens because they are unlabeled. Then we need microtiter plates, which is a 96 wells microtiter plate, as you can see in this image. And then we need a washing buffer solution, such as 1% trifluoroacetic acid. Now, as you can see here, a microtiter plate, also known as a microplate, it is a flat rectangular or round plate with multiple wells or compartments used in laboratory research, diagnostics, and various biological assays. These plates are typically made of clear plastic or glass and come in various formats, including 96 well, 384 well, and 1536 well plates, among others. The most common format is the 96 well microplate. Microtiter plates are widely used in high-throughput screening and experiments, allowing scientists to perform multiple tests or assays simultaneously, saving time and resources. Each well in the plate can hold a small volume of liquid, usually in the microliter range, hence the name microtiter. The next slide shows radioimmunoassay steps. First is immobilization of antibodies. In this technique, specific antibodies are immobilized onto a solid support such as wells of microtiter plates. These antibodies are known as capture antibodies and are specific to the antigen or antibody being measured. Second step is preparation of labeled antigen or antibody. A known quantity of the antigen of interest labeled with a radioactive isotope is added to the well. As already told, commonly used radioisotopes include iodine-125 and tritium. The labeled antigen is referred to as hot or tracer antigen. Wash carefully to remove any unbound labeled antigen. At this point, the radioactivity of the well will be maximum. Next step is incubation and competitive binding. The sample containing the unlabeled antigen is now incubated or added in the well. During this incubation, a competitive binding reaction will take place between the tracer and the unlabeled antigen. If the sample contains specific antigens of interest for binding sites on the capture antibodies, the specific antigens of interest in the sample will bind to the antibodies releasing labeled antigens. Thus, when the mixture of radio-labeled and unlabeled antigens are incubated with the corresponding antibody, the amount of free, which is not bound to the antibody, radio-labeled antigen is directly proportional to the quantity of unlabeled antigen in the mixture. Again, wash carefully to remove the free labeled antigens. The radioactivity of the solution will decrease. So by observation of decreasing radioactivity, it can be confirmed that the antigen of interest is present in the sample. And if the radioactivity remains the same, it can be called a negative test. As you can see here in the diagram, the antibodies, which are the captured antibodies, which are present on the microtiter plate first, are exposed to radio-labeled antigens. And when these radio-labeled antigens bind to these captured antibodies, the radioactivity shown at this particular time will be maximum. Now, if our sample also contains antigens, which are the unlabeled antigens, but are specific to these particular antibodies, a competitive binding will occur between the labeled and the unlabeled antigens, and they will start replacing the radio-labeled antigens and thus decreasing the radioactivity, giving a positive test. The next step includes measurement of radioactivity. The radioactivity of wells containing the bound complexes, antigen antibody or antibody tracer, and the washed fractions containing unbound tracer, that is the free labeled antigens, is measured using a gamma counter or scintillation counter. Hence, the radioactivity or concentration of free labeled antigens is directly proportional to the bound unlabeled antigens. Calculation of results by plotting a graph of radioactivity in percentage versus known concentration of unlabeled antigens, a standard curve is obtained, as you can see in the diagram. From this graph, the concentration of the antigen in the test sample can be calculated. So as you can see in this graph, at zero concentration of unlabeled antigen, this unlabeled antigen is actually coming from the sample. The radioactivity is 100%, means that the captured antibodies have been taken, all the points have been taken by the labeled antigens. And if our sample contains the antigen of interest, which is specific to those antibodies, they will start replacing these hot antigens. And that is why the radioactivity keeps on decreasing with the increased concentration of unlabeled antigen, giving a positive result for this radioimmunoassay. Now let us discuss the advantages of radioimmunoassay. First is high sensitivity. It is extremely sensitive and can detect and provide quantitative results of antigens at very low concentrations, often in the picomolar or femtomolar range. Wide range of applications. It can be applied to a wide range of molecules, including hormones, enzymes, drugs, and specific proteins. It has been used extensively in clinical laboratories and research settings. High specificity. When well-designed antibodies are used, this technique can offer high specificity, minimizing cross-reactivity with other molecules. This makes it suitable for accurate measurement of a specific analyte. Long shelf life. Radioactive tracers used in this technique typically have a long half-life, ensuring that reagents remain effective for an extended period. Limitations. Radioactive waste. This technique uses radioactive isotopes as tracers, which can be hazardous. Proper disposal and safety measures are essential to handle radioactive waste. Specialized equipments. RIA requires specialized equipment for measuring radioactivity, which can be expensive and may not be readily available in all laboratories. Radiation exposure. There is a potential for radiation exposure to laboratory personnel when working with radioactive materials. Strict safety protocols and shielding are necessary to minimize risk. Limited multiplexing. This technique is typically a single analyte assay. Multiplexing to measure multiple analytes simultaneously can be challenging compared to some other immunoassay techniques. Time consuming. This assay often requires longer incubation times compared to non-radioactive immunoassays, which can affect turnaround times in clinical settings. Ethical concerns. Due to the use of radioactive materials, there are ethical concerns associated with this technique, particularly in research involving animals. Radioimmunoassay applications. Endocrinology. It is extensively used in endocrinology to measure hormones like insulin, thyroid hormones T3 and T4, cortisol, and growth hormone. These measurements are critical for diagnosing endocrine disorders. Pharmacology. It is employed in pharmacological research to assess drug levels in biological samples. This is essential for pharmacokinetic studies, drug metabolism research, and therapeutic drug monitoring. Cancer research. It has been used to measure tumor markers like prostate-specific antigens and carcinoembryonic antigen in cancer diagnosis and monitoring. It aids in early cancer detection and assessing treatment efficacy. Immunology. This technique can detect and quantify antibodies against specific pathogens or antigen. It's used in serology studies to assess antibody titers in response to vaccinations or infections. Drug abuse testing. It has been used in toxicology to detect and quantify drugs of abuse such as opioids, cannabinoids, and amphetamines in biological specimens like urine and blood. Fertility studies. RIA can measure hormones related to fertility, such as luteinizing hormone and follicle stimulating hormones. It's used in fertility clinics to assess hormonal imbalances and guide treatment. Research in neurotransmitters. This technique can quantify neurotransmitters like dopamine, serotonin, and norepinephrine in brain tissues, providing insights into neurobiology and neuropsychiatric disorders. Veterinary medicine. It is used in veterinary medicine to measure hormones and biomarkers in animals for diagnostic and research purposes, including reproductive studies in livestock. Food science. It can measure specific components in food such as allergens and contaminants, contributing to food safety and quality control. The next technique we'll discuss is immunofluorescent assay.